Bienvenue à tous. Welcome to Reporters here on France 24. In this edition, the marine research that it's hoped might find a way of saving coral. The threat to the world's coral banks, vast undersea realms of colour and beauty, comes principally from the rising temperature of seawater. But research being led at a university in Saudi Arabia is examining how coral survives in the Red Sea. The high level of salt there should, in theory, make it a hostile place for coral, yet it is there. So, is there a lesson marine scientists can learn and adapt to coral reefs under threat? This is from Alexis Barbier-Boubet and his team, SOS Red Sea Coral.
Seen from the air, you might think this was California. But this campus is in fact just a 2-hour drive from Mecca. The King Abdullah University sits on the banks of the Red Sea. Covering 3,600 hectares and built at a cost of $10 billion, the Saudis have dreamt big. And they've also relaxed their strict rules. This is the first mixed institute in the kingdom, and the wearing of the veil is not obligatory. 80% of the academics come from overseas, like the American Michael Berriman, who heads up the marine biology department. He's a man on a mission.
So the Red Sea's conditions, with its naturally high temperatures and naturally high salinities, in some ways look a lot like what we think the rest of the world's oceans are going to look like in the not too distant future because of climate change. And so understanding all the specifics of how organisms have adapted to those conditions may give us some insight into understanding what the chances are for the rest of the world's oceans to adapt as the conditions change.
This morning, one of the university's boats is heading out on an expedition. On board is the German scientist Sebastian, who's also a trained diver. The team are sailing north up the Saudi shoreline to collect coral samples. The coastline here has not been urbanized, and despite the warm and salty water, the reef is well preserved. An entire ecosystem depends on it. As with coral found elsewhere, this reef is a key nursery for fish and for shellfish. Across the world, 80% of marine species rely on coral. But global warming now poses a serious danger. If the mercury is notched up another 2 degrees, 90% of the world's reefs will die. That's unless experts quickly understand how this Red Sea coral manages to survive the heat. Sebastian now pulls out his hammer and chisel. Coral is neither a plant nor a mineral. It is in fact an animal and a distant cousin of the jellyfish.
Usually I take bigger colonies, so I can only take a small piece from them without hurting them. So the colony, the genotype, the individual itself will maintain in the reef, and usually the fragment we take off should regrow fairly quickly. So it's the least impact we can do to the coral reef and to the population for the corals.
A section is dipped in nitrogen at a temperature of -195 degrees. This sample is for the university's DNA library. The rest is kept at room temperature and will soon be replanted. It's only really at night that you realize the coral is a living thing. It pushes out its polyps to feed, absorbing everything around it, be it plankton, bacteria, or particles of plastic. Fish also come to find a dark corner to hide, because at night, sharks are on the lookout for prey. On dry land, the fires of local oil refineries burn 24 hours a day, every day. Back at the university, Sebastian is now heading out on a dinghy. He's going to tend to his garden.
It's one thing is very long in the center, you should see it.
Just 3 meters from the surface, he set out all of his coral samples. They've been collected from right across the Red Sea. The idea is to spot the cream of the crop, the most resistant to changes in temperature. Sebastian also measures the chlorophyll levels. That's because there are plants living in the coral. A tiny microalgae slips into its cells. It's a perfect symbiosis. If the coral is thriving, it's because the algae is healthy, and therefore there's chlorophyll. While Sebastian nurtures his champion coral, his colleagues are taking a closer look at the algae, which bonds with the coral in seas right around the world. Manuel Aranda comes from Spain. He's convinced that the key to saving the coral can be found in this microalgae called zooxanthellae.
The problem is that these organisms are very sensitive to temperature. And high temperature can induce a breakdown of the symbiotic relationships they have. So under stress, the coral basically expels the algae from the tissue. And what happens is that the tissue now becomes translucent and we can see the white skeleton below. And this is why we call it bleaching, because they basically turn white. And like this, they can survive for some time until they basically use up all their energy reserves they have. And if the stress doesn't get away, then they start to die. They basically starve.
As soon as you touch this seaweed, the coral dies. So how can it be studied closely? Manuel has found a solution. He uses anemones. They survive thanks to the same symbiosis and are much less sensitive.
So all the red dots that you see here are actually symbionts that are sitting inside the cells of this anemone. It's basically the same algae that live inside the coral tissues, basically the ones that provide most of the food to the corals. And they live inside the cells. So you cannot be more intimate than that. So it seems to be kind of a codependence that they have evolved over millions and millions and millions of years, more than 140 million years. So it's a very, very ancient relationship.
These cabinets are anemone incubators. Each one is set to the temperature of a different world ocean.
And we have the strains, the same anemones, from different parts of the world. We have them from Hawaii, from Florida, and from the Red Sea. And these are places with very, very different temperatures. We can actually bleach them and keep them without any symbionts at all, and then put in whatever symbiont type we want. Because the algae that we call Symbiodinium is actually very, very diverse. There's many, many, many different types of these algae that are all very, very different. What is it that makes temperature resilient? Is it actually the host, or is it more the symbiont that they kill? And these organisms allow us to basically plug and play and exchange the symbionts between the different host strains and see what happens.
By using trial and error, Manuel creates the best fusion between the algae and the anemone, and then applies it to the coral. It's a simple selection process that any gardener or breeder would do. Others, however, want to go further and actually access the coral's DNA to modify it. Many ecologists are against this idea, fearing it could set off a negative underwater domino effect. For the head of the department, there is no such taboo.
You know, unfortunately, we may be pushed into a situation where we have to start thinking about even more proactive solutions. That, you know, can we engineer corals? We have to deal with the questions of should we later, but let's start with the core scientific question. Is it possible?
While scientists ponder over genetic modification, the selection process continues in earnest. This is what's known as brain coral. Each section comes from a different region. For 6 months now, they've been getting accustomed to each other, as any animal species would. And now they're ready to reproduce.
For our experiment, it's important that we see what happens if we interbreed them, if we cross them. How does the offspring perform?
The corals from the south, which are used to much higher temperatures, if we can basically take those adaptations that they have and inject them to the corals in the north, so that we can basically create coral populations in the north that will withstand temperatures that they might see, let's say, in 100 years from now. And with this, if we start early enough, we hope that we can start building more resilient populations of corals along the Red Sea.
The reproduction will occur at sea.
We can transport it easily without having to worry too much about it to the nursery, where we would place them and let them acclimate for the next spawning season.
The spawn will be triggered by a full moon in a few days' time.
Corals have survived so much. They have survived so many mass extinction events on this planet. They've lived through phases that were much hotter, that had much higher CO2 levels. But if we look at these times, what we see is that the reefs are gone. Basically, they're gone for millions of years, and then suddenly they come back. So we're not afraid the corals might go extinct. So the research that we're doing is not to save corals from extinction. Climate change will kill a lot of species, yes, but I'm pretty sure that they will come back. The problem that we are facing is that we're going to lose the ecosystems they have built. We cannot afford to wait hundreds of years. We cannot afford to wait thousands of years for corals to come back. We need them now, because now it's when they feed hundreds of millions of people on this planet. It's now where they provide the home, the habitat, for many, many other marine species.
500 million people live off our oceans, and coral bleaching would have an immediate impact on mankind. Sebastian and Manuel, the two marine biologists from the King Abdullah University, are busy creating a colony of brain coral. If all goes according to plan, this colony will be able to resist a further rise in temperatures. This super coral would allow the Red Sea to preserve its breathtaking beauty. That would definitely be a piece of good news for all of our oceans.
Our film SOS Red Sea Coral from Alexis Barbier-Boubet and his team. You can see it again via our website, france24.com. This is Reporters, France 24. Stay with us.